Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we are thrilled to announce that our ecosystem is growing and are delighted to count Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and Montana Molecular as our ecosystem partners. Become an ecosystem member yourself and join our partners and your colleagues today. The ecosystem is your GPCR-focused virtual playground. Join over 400 of your peers who have already started exploring, connecting, and collaborating better. You can explore the ecosystem by signing up and getting a free site membership. For the next couple of weeks, once you sign up for your free account, you can watch the talks from the third edition of the Dr. GPCR Summit for free. When ready, you can also get a paid membership to unlock the ecosystem's full benefits. If you'd like to register your team or company, or if you live in a developing country, please reach out to us by email or on the ecosystem, and we'll be happy to help you join us. The list of benefits of the ecosystem is quite long, but today we want to highlight that you can get direct access to our ecosystem partners, ask questions, and who knows, find your next job in the ecosystem. Our job board is a GPCR-focused one where you can explore different opportunities, and if you are looking to hire, you can submit your job description. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Explore the possibilities by navigating the site using the direct links in the footer. Check it out today at ecosystem.drgpcr.com. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I am delighted to have with me Dr. Sri Kosuri. He is CEO and co-founder of a company that I heard about earlier this year, and I'm super excited to learn more about Octant. Hi, Sri.
Hi, Yamina. Nice to meet you.
Nice to meet you. So excited to have you on the podcast. So let's start at the beginning. Please tell us, what do you do and who you are?
Sure. So my name is Sri, as you introduced me. So I'm co-founder and CEO of a company called Octant. We're here in the Bay Area in Emeryville, California. What I do on a daily basis is help run the company. I think I have kind of a previous life in science. I was a professor prior to this at UCLA, and tangentially related to GPCRs. I'd say I'm a GPCR fanatic more than I am a deep scientist or fan even of GPCRs. I think they're fascinating receptors. But yeah, in general, the company has been going since 2017. I shut down my lab in 2019, and when I went full time to the company and moved up here to the Bay Area. And yeah, I've been building since. The company is about 60 people now, so it's been growing quite a bit, especially over the last year or so.
That's fantastic. I heard about the company in March where I was at a virtual summit, and I heard it talk about it, and I was like, oh my God, this is so amazing. I have to talk to you and I have to learn more about it. But before we get into that, tell us a little bit more about your previous life as a professor. And let's go further even back. Did you always want to be a scientist?
I think growing up, yeah, I was always pretty interested in science, and I'd say engineering as well. Like I love like reading manuals of VCRs, if you remember what those are. And,
I do.
I liked kind of math and engineering and science, and I couldn't decide on a major. And so I actually joined Berkeley as an undergrad as a what's called an engineering science major. So I couldn't pick between the two, and so I picked that. And then my junior year, I was always interested in biology. And in my junior year, this new major was starting called bioengineering, and I transferred into it in my junior year and graduated, I think, as the first bioengineering major at Berkeley. And so, and then, yeah, so, but I guess my love for research really started maybe my beginning of my sophomore year. I kind of met this. He wasn't quite a professor yet. He was kind of a staff scientist at the Lawrence Berkeley National Labs, named Adam Arkin, and started working with him. And I really fell in love with research, and I just spent almost all my time, like in undergrad, doing research in his lab. I was all computational at that point, so I was looking at stochastic gene networks or noise in biochemical systems and how it arises. We studied like kind of phage development and bacterial development kind of processes. And that was really fun, and decided to go to grad school. So, and then I went to grad school at MIT, also in the nascent new department called biological engineering at the time, or it wasn't quite a department then, it was just a grad program. Um, and I joined a lab, kind of doing similar types of things. He was the chair of the department, this guy Doug Laufenberger, but then a new faculty had moved in, who I'd known while he was at Berkeley previously, this guy Drew Endy. And when he joined, I kind of got more and more excited by what he was working on. It was like I was looking at kind of phage development, and I decided to join his lab. He was a fellow in the biology department and then became a professor later. But so I just switched labs at that point and started working on that. And then kind of maybe 2 years into that, I was building these computational models, got sick of that, didn't really believe them, and started my like long foray into experimental biology. And so starting with kind of working on building basically synthetic viruses that were more like our models, and then really starting kind of work in this field of that was just starting called synthetic biology now. And
Yeah.
so building viruses. Then I went to a company after that. So I went to actually a venture company. We kind of founded a startup trying to make algae, make biofuels back in
Nice.
2007. And then I was there for a couple of years, and then I decided to come back to work with George Church. They were starting this new institute called the Weiss Institute, uh, where they were trying to bring back people from industry. And so I started there kind of as a postdoc and then staff scientist, working on DNA synthesis technologies, DNA editing technologies. And then what turned into a lot of my lab, which is building ways to do large scale experiments using sequencing as a readout. Can talk more about that if you want. And then I started a lab at UCLA in 2013 as an assistant professor, and then and got tenure and built a lab. And then a couple of the projects that was started nascently there were in GPCRs, and we got more and more excited about that. Decided to start a company in 2017. I just mentioned that.
And
Moved
here we
full-time
are.
company
Well,
2019.
it's so interesting. Thank you for walking us back through those days. What comes through to me based on what you were mentioning is that it's high throughput, high level systems thinking and studying systems. And I think the computational aspect that you mentioned, maybe it's just me, but I think maybe you were ahead of your time at that point, or your interests were ahead of your time. I feel like synthetic biology and all these computational methods are, you know, people are using those in companies, but also in academic settings to study all sorts of questions.
Yeah, I think reflecting on that, like I think it was, I think maybe the way I think about it now is ultimately like all of these cells and cell lines and whatever we're using are all just models of some physical system that we're trying to understand. And what's amazing about biology is like if if you had ways to like change the things you wanted and then like measure them output, you can do millions of experiments in ways that you can't really do in any other physical system. And like what biology can do is so amazing. And so the way I think about all of these approaches is really like trying to do experiments at scale, like basically using the cell models that you build as like almost simulators of what you want to do. And the inputs are DNA and the engineering that you can do, and the outputs are the readouts that you can read out. And can you use this to understand and engineer those systems? And that's still kind of my thinking, but it took like, I don't know, 15 years to build out the suite of tools to do that kind of work at scale.
I think so too. I think it's I like thinking about problems at scale, and really you mentioned NGS and sequencing and having all these tools to manipulate a system and then acquire a ton of data and then get as many answers as possible within the same system. I think it's a kind of thinking that I wasn't I didn't grow up in, and I think the audience who's going to be listening to this podcast is mainly one receptor, one ligand. We have hex cells and we measure some things in there. That's why I was fascinated by the talk I heard at that meeting about the capacities and the type of assays that you can run at Octant and measuring hundreds of responses in the same plate, which I think is just phenomenal to be able to do that. When did your interest in, I want to say, GPCRs come up, or where did you first hear about GPCRs?
Yeast. So I like definitely
Yes.
there were yeast like pheromone response and things like that. The first time I heard about them, but I started getting really like my entry into GPCRs was super random. Like basically, I just got really obsessed with olfactory receptors for a while. Just
I
reading about
like.
it. And from the engineering side point of it, it was just like, oh, this whole system is amazing. Like you can sense all these chemicals and at like really insane precision and be able
Yeah.
to tell them apart. And so I started getting really interested in that. And I kind of on a lark wrote a grant on this idea to build like basically a multiplex assay. Like we worked on different ways to do these kind of multiplex. And maybe I should back up a little. Like my lab would work on like individuals in the lab would kind of think about a new way to look at some new system, whether it's like protein interactions or splicing or gene expression or
Yeah.
whatever it was. And like how do I convert that kind of assay into something I can control with synthesis and read out by sequencing? And so you could imagine instead of a classic luciferase assay, you can output a barcoded RNA. And as long as that barcode tells you what you're looking at, then you can do this. You can do this change of like I can make millions of changes and then I can like read them all out all in a single dish, right? By one student if they can like go through all of that. And so we did that across a lot of things. And one of the things that fascinated me is like, could we do this with GPCRs, especially olfactory receptors? And really naively, I wrote this grant to this place called the Defense Threat Reduction Agency. And I just wrote it. And it was like, I got it. I got a notice of award. It was like a huge grant from my lab. And I'm like, oh my God, we need to start doing this. It's got this notice of award. And so, and one of my first grad students, this guy, Eric Jones, was like, yeah, I want to do it. Right? So we started working on it. And then we started seeing some interesting early signs of like might work. And then um, but like every month went by and no checks were arriving for the grant.
Oh,
And like I kept on like I was like, what's going on? We got this notice of award. When's it going to start? I tried trying to get the program up, so I couldn't. And then eventually, like I got a hold of someone who said, "Oh, that program officer was fired, and all the programs were cut,
oh my God.
and like your grant doesn't exist, right?"
Oh,
And I'm like, "Oh my God!" And the university lets you spend once you get a notice of award. So I had spent all this money, and I had some startup and so I'm like, oh God. And then this other grant opportunity from the NSF came along called Ideas Lab. And I don't know if you ever heard about that, or it's called something else now. But it's basically like Hunger Games for scientists. It's like
okay.
you show up at like we had to show up at Genelia Farms. All the reviewers were there, and all these new people are there in the field of olfactory receptor, like all these, like, but also like random people like me were also there. And the idea was like come up with some new idea together in groups and we'll fund like three of them.
Oh, wow.
And like six groups formed, and everyone's competing at like no one's sleeping, and it was really fun. But I met a number of people, and especially Hiro Matsunami, who helped us. Like, and I reached out to some of these people as we were kind of looking at olfactory receptors, and so that's kind of how it started. So we were starting on olfactory receptors, and it's like hard going, like expressing olfactory receptors, getting them to functionally signal in hex cells. It's like
Yeah.
impossible. Like we were,
Yeah.
and so Eric had been trying all these different ways to get them to work, and nothing was working. His committee was like, "You need to find something else," um, and I was like, look, just put a bow tie on it. Like, just finish all the last experiments you plan. Like, put them together, try all the things. And if it doesn't work, at least we have this thing of like all these things didn't work.
Yeah.
And like, let's start thinking of a backup. And at the same time, I was at Duke, and I had met with Hero, and I asked to meet with Bob Lefkowitz, and I was telling him about this project, and he's like, "That's super cool." He's like, "Don't work on olfactory receptors. What are you doing? This is super cool. Just work on beta two. It'll work like gangbusters." And I'm like,
Yes.
and like, and like, and so then, and then I brought that back to Eric, and Eric was like, "Yeah, let's just try it," and that worked immediately too. But the problem was that his bow tie experiments started working as well.
Oh,
So both experiments started working at the same time, and like, but Eric, being Eric, and he's awesome, like basically was like, okay, we're just going to do both. And that's kind of how we started on this thing. So the olfactory thing was like, oh, we're going to do a lot of different olfactory receptors in a single well, and then we can put drops of like aroma, and like we had
Yeah.
super smelly lab, and like it was like, and that was the idea. Then the thing with the beta 2 was that, oh, well, we can also do stuff like, let's make every possible mutation to this receptor and read out activities like, cyclic AMP or other types
Yeah.
of activity. And that's like stuff my lab's good at. We build like DNA from microchips, and we can put them and barcode them and put them
Yeah.
into human cells in different ways. So all of that stuff is stuff we're really good at. The GPCR stuff was new, and um, the and so we started like working on those projects, and both of them started looking like they were working, and like we had like a bunch of really early data on. And so that started getting exciting, and then we started thinking really about like, what does this mean? What could you do with something like this? How do we think about these types of technologies and how they might impact therapeutics?
Yeah.
And it might so that's kind of the origins of a lot of this, right? And I think GPCRs are obviously commercial in many ways, and
Yeah.
uh, a lot of this was just really learning about the field, and then once starting the company, really learning about the industry, which is very different than the academic field, right?
It
And
is.
so I think that's been just a constant education. And we don't only do GPCRs at the company now, but like it is it's kind of how we started into this world of therapeutics.
Well, it was a GPCR conference that I went to, so hence the talk was about GPCRs.
Yeah.
So interesting that you picked olfactory receptors. I mean, you picked one of the hardest classes of GPCRs to work with. Although I agree, they're fun, they're very difficult, and they're not like the beta 2 adrenergic receptor. Where did the idea, like, how did you even think about olfactory receptors?
It kind of came from we had been doing some bacterial biosensor. So I had been part of some work to like trying to sense natural ligands that are coming out of in like synthetic biology. Like how do you sense like molecules that you're
Yeah.
making, like use it for and I think everyone knows smell and how it works. And so I started just looking into it, right? Like how does it work? What are the receptors? Like how are insect receptors different than regular? Like, and so like
Yeah.
I think that was like it was really just that. It wasn't anything smarter than that. It was like starting to just dive into the literature a little bit. So I like I thought it was just as I read more and more, it just seemed super fascinating as a problem. I was a young professor. I wouldn't have had the guts to actually do it if I didn't get a random grant that never materialized.
Well,
like I was just like, I don't know enough about GPCRs to like launch this project, so I'm not going to do it. But I'm just going to write a grant to see if like I
yeah.
can get some like money to do it. And it's kind of like if I never got that defense grant that never actually materialized, I wouldn't have had Eric like start that project. And like a lot of it's kind of random, right? Like it was just one of these things. And
But still.
like it was a little bit of a series of unfortunate events or
Which
fortunate
is great because I think you made a great point by thank you for sharing that story. I think it's such a fun story, and I think everyone who's listening to the podcast is going to really have fun. But I think the take home message out of that is that just follow your gut. You're interested in something, just find a way to learn more about it. And you sat down, you wrote that grant. It did take you time, and yes, you never got the check, but at least you took a chance. You were interested in something and you did something about it.
Yeah, I think that's right. I think as like a new professor, it's always like, do I do something completely different, or do I do something like that I know that'll work? And it's probably some portfolio of those things. And um, yeah, I think for me, like grant writing is like a thing of like consolidating ideas and trying to say like, is there a path here that makes sense, and how do I
Yeah.
think about it? So I I think using grant writing as that exercise is really fun. I think a couple of like really of the cool projects that came out of the lab started that way, where it's just like, this is an interesting problem. Let's see if we can write a grant on it, and see what would happen.
I think that's fascinating. So what happened when you went for that? I can't remember the setting where you had to form groups and compete
Oh,
in
the
a
ideas lab.
yeah.
So yeah, so I ended up being a part of a funded group. Uh, with like a whole bunch of like Lisa Stowers and Hiro Matsunami and like a bunch of really great GPCR biologists. like in vivo physiologists, and we had this whole thing, and we had a couple of papers out of that, but like it just started to like the beta 2 paper kind of was separate from that, and like um and because it had nothing to do with olfaction,
Of
but
course,
like
yeah.
it just got us into playing around in the space and like understanding the possibilities that might be possible and looking at this interaction of like small molecules with receptors, and can we start deconvolving it. I think the heady, very naive idea was like, look, we can build reporters for all sorts of things, not just olfactory receptors or not just
Yeah.
GPCRs. Um, like there's only 20,000 targets in the human genome, and there's more than that many cells in your well. So like, like why can't we start mapping more systematically how molecules are interacting with the targets of human physiology, and how do we think about that? That's kind of like I think the bigger stuff that came out of it, but the grant and the it was really fun. It was really fun to work with a really accomplished crew of GPCR and in vivo biologists in olfaction. I think it's a super cool problem. It's a very long term problem, very clearly, but
It
like it's a
is.
very cool problem.
It is a very cool problem. So how many days was this competition? How many days did you
It
not
was a
sleep?
week. It was like I didn't sleep that whole week. You could, it's really fun to talk to. um, yeah, it was really, I actually thought it was really interesting and fun, and like much better than some ways writing a grant. Like the reviewers are right there, they're like
Yeah.
like giving you a hard time, like, and and so I thought it was really interesting. I thought it was fun. But yeah, there was very little sleep that week.
I love the idea of, having grants reviewed in this way because then you get your feedback right then and there. And I think it would make the whole process a little bit more dynamic.
It was-
Stressful, granted,
yeah,
but more dynamic.
yeah,
At least you get your feedback right there and then.
yeah. I think it was interesting.
So, any
Um,
take-home messages out of that experience? What did you learn about GPCRs or about your ideas or your working style after that whole week of interactions?
um, I I learned that like I don't know. I learned that like having kids and being able to operate with very little sleep is an advantage. I
Yes.
mean that was a it. I don't know if we learned a lot, right? It was an effort in like how do you combine a set of people towards a thing, right?
Yeah.
Like it's a bunch of professors that have their own like research programs. They all think they know what they want to do, and you're like, okay, we have to come up with something, and none of us would do otherwise, right? Like,
Yeah.
and I think to say like how successful was it? I think in some ways, like, did we exactly accomplish what we set out to do? Probably not. No, I think like if the answer is like, did it drive a lot of create? Like, I now quit tenure and moved on to a company because of like that
Yeah.
stuff. So, and like I think we're doing exciting things that are good for the world, and in that sense, I think
Mhm.
that's like a really great outcome. So
Do you
I don't
think
know.
do you think that week fueled your ability to, work with different people from different backgrounds, one science wise? Did it
No,
help
I
you?
think I've
No?
always been like that. Like I think I am a big proponent of like learning languages in all these fields and like spending time. Like, I remember as like a new like in the church lab, you can just sit in the church lab and have a language, but you can also go to the Department of Genetics talks. You can go to the like
Yeah.
the biochemistry talks, or right down the hall. Like like being able to speak those different languages, I think for me as a tool developer has been really really important to understand like what are real problems, like what are not real problems. Uh I don't think that came from like a week,
Mhm.
right? That's like
Yeah.
definitely spending the time trying to like be able to speak in the languages of these fields, right? Like if you want to talk to a GPCR biologist, you kind of have to know some set of languages to have a high or vocabulary to have
Yeah.
high bandwidth conversations. And I think for like people that are younger in their careers thinking through this, I think that's a super important skill to try to learn.
It is, and I think it's something that, unfortunately, a lot of times when you're doing a PhD or a postdoc, you're just so, especially if you're doing wet lab experiments, then you're so focused on that little thing to make it work so that you can finish up your project that sometimes you don't get that opportunity to look up and go to different types of talks and talk to different people and learn different languages and potentially apply tools used in other fields to your problem?
Yeah, I kind of got away with that. I kind of was forced to do that because I've always like in my lab, I was like the first experimentalist, right? Like my
Hmm.
PI was largely computational, and same with like, and so like we had to like learn all these languages by ourselves. Like we kind
Yeah.
of had to run around. Like we actually started a whole website called Open Wetware. It's still around. It's like a huge wiki with thousands of labs on it. And like, basically the idea was like people should write down all the stuff that no one tells you in protocols because like it's
Yeah.
nowhere to be found other than like passed down by lab by lab by
Yeah.
lab. And so
The trick of handling the pipette in this way because the
yeah.
stars are aligned that
Oh
way and
god,
this
there were
way.
so many of those things. And so like yeah, so I think I think having that experience of always being like an outsider in a field is like I don't think I even have a field. Like was a professor in chemistry, which makes no sense because I know like very little chemistry, and like bioengineering is kind of a mutt of a major as it is. And like so I for me it's been good. Like I really do love and appreciate people that have very deep expertise in one area, and I think but like I think there's room for all types of people,
Yeah,
right? And so I think
agreed.
it for me
Yeah.
it's been useful.
Yeah, I'm not saying that, one thing is bad and the other one is good. I'm just saying that sometimes it's absolutely worth looking up and looking around and figuring out what works for you as a person and as a personality. You seem like the type of person who's really driven by solving problems, and you're looking at building tools to solve those problems. And sometimes the problem can be interchangeable, but as long as you can think about building a tool to help answer some questions, that's your driver based on what I'm hearing.
Yeah, a lot of it has been like a lot of my career has been that way. And like and then like I think it's important to learn the languages and like the problems because like I think the biggest fear I have is like no one uses what we build, right? And so like
Exactly.
I think and it's not useful actually. And so like I think it's important to like stay close to the problem and so I think there's it's hard to do both, right? And so
It is.
there's some side of the slope that you're on in
Yeah,
some ways.
it
Yeah.
is. But I think you have to you can also be that person who can adapt
Yeah.
and dive deeply into a problem in order to see if the tool is going to work out, or build a tool around that, and then get out of there and go into another problem, learn a different language and build another tool, or see if the
Yeah,
same
and
tool
so like
applies.
and you sometimes find opportunities or sometimes they come to you, right? Like we in the we had just moved up in 2019 and the pandemic hit in early 2020 and we had built this back end of our platform that, in each well, we're doing this barcoded RNA-seq essentially, uh, of a particular set of amplicons that are like
Yeah.
measuring our reporters, and and ultimately, like we made that really cheap and high throughput, so like individuals could run the whole thing without much automation with thousands of wells and like
Yeah.
or like $1 a well basically, and we knew this would work. We knew it was quantitative and then COVID hit, and we're like, "Oh, we could turn this into like a
Mhm.
COVID sequencing thing." And like, it took like 2, maybe 4 weeks to show like we went into the lab as everything shut down
Yeah.
and like showed that it could basically detect COVID amplicons and like all this kind of stuff, and like we could do other amplicons. Um, and then we released it open source, right? Like we kind of like signed the Open COVID pledge. We like talked to it, and it ended up like it took. But like I think the thing that's much harder to see is like the next year or years was just like getting that like through FDA approval and through like
Yeah.
CLIA and through like and actually building the infrastructure and actually delivery and reporting and all like that takes so much work and,
Yeah. Yeah.
and so like we ended up like UCLA ended up adopting it. Like they like their whole campus is tested by that technology, like in Caltech for a while and UCSB. And so it was cool to see like a small idea turn into it. But then there's also the you know
The other
like
aspect of, yeah.
95% of work that you have to do past the initial kind of demonstration that is the hard part. And I think
Mm.
that's also
Which which one do you like? The 95% or more the discovery side, the proof of concept to ensure that, it's the technology is working, it's
oh
inexpensive.
yeah, I think it's the latter. Like it's like it's the fact that like 2 million tests almost have been done at UCLA, right? Like, or I think it's north of 1.5 million, right? Or like whatever, like multiple big organizations have like companies have taken it on, like that is actually in use is like what's satisfying to me. Like
Yeah.
that has been some of the most satisfying things in my career is like we built this thing that came like the 2 week idea is fine, it's cool, but it's not like it's very unfulfilling. And I'd say like 95%, like for me, writing up a paper has never been super fulfilling. No, writing a paper is fun, but like where it goes or where it gets published or like
Yeah.
how it like is not the not something that has all like really motivated me over time. It's really like, is this thing like actually going to make a difference in the world? And so it's much more the latter, but like that is just a lot of hard work,
Yeah.
right? And so yeah,
It's part of a process, and you have to go through all the steps in order to, mention FDA approval and all of these things. It has to be done.
yeah.
Getting back to GPCRs, actually, before we get back to GPCR, I always ask this. You may or may not have a favorite one, but before we do that, you're a professor, and then you went into industry. How was that transition? Like, was it a difficult decision for you to make? I mean, tenure is not, not everybody gets to get tenure. It's a difficult thing to get. How did you make the decision?
So one, I think for me it was less like I've been in industry, like I went after grad school and then
Yeah.
came back. I've been on SABs. So um I think I thought I knew what it was going to be like. I don't think that was been true, but I at least at the time of decision making, I thought I knew. Uh I think I think there were a lot of factors. Like it wasn't any one thing, but I think from like the pull side, I was just really excited about like building a team around this stuff. I really like building. I was like the first person in my undergrad lab. I was the first person in my grad lab. I was the first person at the company that I helped start. Right, I was the first person at this new, one of the first people in this new institute.
Yeah.
Then when I started a new lab, I like that process of building. I think when you get to around tenure time, like I don't know, for me, it felt a little scary, right? Like it was like, oh, I'm going to die in this office in 30 years. That's my like life. Uh,
Buried under papers.
yeah, like I just seemed fun to try to do something. I think it was also like convenient. Like I knew I could get all my students in a good place and like get that thing happening. And then it was also like I was co-founding it with a friend, and um I think there were those things. I think there was the latter thing was also like making sure that I was working on the right problems. Like most, like I had never been in discovery, and as it was moving more and more into like human translation stuff, I just felt like I wasn't working on the right problems that is actually going to impact therapeutics, and I just felt like I wouldn't be able to learn that in academia very effectively. I think that was one, and then two, um, yeah, I think like there's lots of reasons to do it, but like I think it's fun to build. Like I think my career has been in these like kind of 10-year chunks, and I think it felt like the right time. My kids were not super young at the time anymore, so I felt like I could like really focus on building something new. Um, I I think those were
Mhm.
the kind of
Thoughts?
real thoughts. It wasn't like I don't have a lot of sunk costs kind of idea. Like oh I built like um I feel like I can always get a job somewhere. Someone would hire me doing something, and I don't have a lot of expensive tastes or anything. So like I still wear
Yeah.
free t-shirts mostly. And so like I
Take the
feel like
best.
I worst case scenario is it doesn't work. And we took a swing and like it
Yeah.
didn't work. I'll probably have learned a lot. And uh like who else should be like no one else is gonna build Octane, right? Like it's not like we were going after the same thing that everyone else was going after. We're like, you know, we're not the 35th gene therapy company going after
Yeah.
like,
The same
like
gene.
sickle
Yeah.
cell. Yeah. So like no one else is gonna build Octane. It was kind of a crazy idea. And so like it
Why
felt
not?
then to just go do that. Right. And so like that's kind of what the
I love
uh,
it. I love it. I think you made a lot of great points, and let me recap a little bit for the audience and for trainees, because I think it's important to think about it. Yes, you do have a set of skills that allow you to find a job if it doesn't work out, which, you know, I don't think it's going to be the case, but at the same time, you had to try it. So if you're faced with the decision of trying something and building something new that has a purpose and serve a higher purpose and help people, why not? And you seem to be having a lot of fun doing it and learning new things while using the skills that you already bring to the company. And I think that's important for the listeners, especially the trainees, to think about. There's no linear path once you start a PhD. You can do many things with a PhD. You can become a professor and then decide to build a company, or directly go build a company. You never know when you're going to end up, but the only things that you might regret is the thing that you don't do.
Yeah, I think that's right. I mean, I think like you also don't want to like there's the opposite end of that where you're trying something new every year, right? So like I think there's always a balance, right? But
Yeah.
yes, you're right that most people probably probably think too much on the safety side.
Yes.
Um, like if
Yes.
you're like there's lots of things to do, and that's the other thing is my dream was never to always be a professor, right? So I feel like I could be happy doing lots of things. And having seen industry like the first time I was like went into industry, I definitely had people who were like, "Why would you ever go to industry? There's only like you seem relatively smart. Like, why would you do that, right?" And like, and then you get to industry and you're like, "Oh, there's all these really smart people that are really earnest about changing the world, and like that's amazing too." And so
Exactly.
like I think there's I think once you start getting outside of like that, then you start realizing that like all over the world, there's like really earnest people trying to do their best
Exactly.
and there's a lot of fun things to do.
Yeah, and it's interesting you mentioned, risk averse. I was risk averse for a very long time, and in my mind, it was, master's, PhD, postdoc, professorship, and then it didn't turn out to be that way, and I ended up in industry at a startup, and it was so much fun building something. You have a blank canvas, you have a lot of deadlines, it's a lot of work, but you get to build something really cool. And out of that experience came the podcast, and we're 2.5 years in, and I think today we're recording episode 94 of the podcast, which I think
That's
it's really
awesome.
awesome. All right, cool. What is your favorite GPCR, if you have one? Or it can be any other GPCR related protein.
Oh man.
Tough one, right? It could be a family, but actually we kind of identified the family already.
We did this thing, and maybe Naomi talked about it, where we took all the aminergic GPCRs,
I
the
think she did.
35 or so receptors and like their different couplings and like kind of multiplex them, and then ran things like atypical antipsychotics through them. Like the complexity of how small molecules interact with that class and like their impacts on physiology makes absolutely zero sense to me. Like it's just like the whole problem is so weird. And it's super fascinating, right? Like these things have huge impacts in physiology. Slight changes in like chemical structure can lead to massive changes in physiological impact. And it's like playing these like there's some like set of chords being played and like you're not sure what notes are going on, and so I think it's I think that's super fascinating. I think the whole problem is really like I think olfaction is very similar to that, but I think it's very different when you see these complete like it's one thing when it's like two different smells. It's another thing
Yeah,
when it's like, oh, this
hundreds.
is like going to make you trip, and this is like a good anti-anxiety pill, right?
Yeah,
Like, like I think, I think that's fascinating, and like some of them are blood pressure medications, and some of them are going to be anti-asthmatics, and some of them are going to be like it's so it it's completely fascinating. I think that's fascinating. I think I have a lot of really ones that are kind of high on the list. I think um the chemokines are pretty exciting and interesting too from that kind of same angle. We've been doing a bunch of stuff in a class of receptors called the melanocortins, and that's been
yeah.
uh, also like just looking at hypothalamic signaling in GPCRs, and the neuropeptides are really super cool. I don't know. I have a lot of favorites, so it's
which is
hard
great,
hard to,
which is fantastic.
hard to track down.
I'm listening, and
Yeah,
in my mind, I'm like, yes, oh, that's cool too. Oh, that's cool too. And
yeah,
I think you mentioned a small molecule that's going to be an antidepressant versus going to make you trip, and I find not only the concept or the this idea of having changes in molecules and have a different set of responses in behavior, but I'm thinking about every time. Okay, maybe this is too much information, but long story short, I was on the operating table and the anesthesiologist was giving me stuff, and everything that he was giving me to regulate blood pressure and all of these things, the only thing I can think about, this was not the right time to think about, is like, okay, so which GPCR does it act on? And the effect, the worst thing or the most phenomenal thing is that it was instantaneous. He
yeah,
pushes the thing in and you feel it. And I'm like, okay, so we've activated a G protein, we're signaling, we're doing the right effect, and everything is hunky dory in the system. And I think this is just phenomenal. And I don't know if you've seen, but Brian Ross lab published the LSD, the structure of the GPCR with the LSD, and I was watching the video, and honestly, I had tears in my eyes. You could see the molecule in the receptor. It's like, wow, so you're tripping because of that little thing in there that is
yeah,
actually occupying that space, which I find really, really fascinating. So I
yeah,
guess the answer to my next question, do you think GPCRs are still good drug targets, is definitely a yes
yeah.
on your end.
Yeah. I mean, absolutely. I think it's a really interesting and complicated problem. And I think we have a very long way to go to figure
We do.
it out, right? Like,
We
I
do.
still think it's going to be a very long time before we have even close to all the answers in this receptor class. And I think like small molecules are like, I know there is like both my field and my history is more in this like gene editing and all this kind of stuff. And
Yeah.
like, but I think ultimately small molecules are just kind of really magical, and the ability to like alter like specific biochemical mechanisms is just becoming increasingly possible by all types of small molecule chemistries. And that combination, I think, GPCRs clearly affect physiology in all sorts of ways. And if we can figure out the right ways to like hit the right notes, then that's super exciting, right? I think it's a hard problem. I don't think
It's a
that's very easy, but like I do think there's a lot of potential.
I think so
Um,
too, and I think we've talked about small molecules, but then the next class of tools that or therapeutics that people are working on and thinking about is antibodies, and I think that takes it to the next level. Because as you know, there's some efficacy and sometimes issues. I'm sorry, it's the end of the day here, and I'm having a hard time spitting out what I want to say. But basically, having different classes of therapeutics, whether they're small molecules or antibodies, really helps. I want to say broaden the number of molecules or any type of therapeutics that we can look at to modulate GPCRs in the therapeutic
yeah, I
context.
mean, I interestingly, I'm not super bullish, right? Like, I
Okay.
don't think, I think there's a few examples, right, where specifically you need to like inhibit or even activate this
Yes,
very particular receptor to cure disease. I don't think there's many examples where that's super clear. And
and
like,
that's the problem.
secondly, like, um, maybe CGRP or something, right? Like, but there's not that many. And then, um, the magic of small molecules is that you can do a lot of different things, right? Like, can you alter, you know, can you build a bias ligand? Can you like alter trafficking and or like
Yeah.
recycling? Are you like hitting multiple receptors in that class? Can you act as an inhibitor to one and an activator to the other? Like, I, I think I think that kind of thinking, especially in GPCRs, is probably going to be important over the next 20 years. And I think that's largely still, unfortunately, or maybe fortunately, because we're getting better at programming small molecules, like going to get easier or going to be the way that we're going to see a lot of new biology. That's my personal kind of
I
view on.
like I like I like your view from the perspective of the fact that we have done so much work on small molecules. We're good at chemistry now. We can modulate our molecule. We can, change them. We have the systems. Um, most likely it's easier to work with small molecules compared to antibodies. But and you're right in the sense that when you mentioned that we don't have a clear understanding of, for disease A, we have to have an agonist for GPCR B in order to induce this effect and modulate the disease outcome. That's a black box that we don't know anything about. And small molecules have been it's easier to program these, and you have an agonist and antagonist, or an allosteric modulator. I think it's more difficult with antibodies. Antibodies have been, when people think about antibody therapeutics against GPCRs, they think about inhibition mostly, very rarely about agonists or modulating these functions. But I think it's also a matter of not having the enough tools or the right tools to generate many antibody therapeutic or antibody binders that will do all of these functions. I think it's two sides of the same coin. On one side you have the small molecules, on the other side you have the antibodies, and as long as it becomes a therapeutic to help people, it doesn't really matter which one works better. But each side of the coin has its difficulties.
Yeah, no, I'm trying to point out is like uh I think the big gain you get from antibodies is specificity and safety, right?
Yeah. Yeah.
So I think the safety side of that is right, but I am wondering if the specificity is not the only aspect of it, and I think there is a general thing of like, yes, we need like a super specific GPCR, and I think that might be true in like certain indications, but like if you look at like kinases in cancer or things like that, but like I think for GPCRs, actually the lack of specificity is a really powerful thing for some of these small molecules, and to enact really powerful phenotypes in humans. And so, like, that's all I'm saying. And
Yeah.
but I think we'll see. And like, yes, like it's good that there are many bets being taken into the world.
Exactly. And you mentioned, the lack of specificity. You don't know. You might have a good molecule or a good antibody that does something in your test system, but until it ends up in a human, you don't know what is going to be the long-term effect. I know we're running short on time, so let's skip to the next question. We've talked a little bit about, your career and the perspectives that you've taken. Any advice you might have for junior scientists when it comes to contributing to the field, but also to contributing to the greater good in general with science?
Yeah, I mean, I think there's like the boilerplate stuff to say, right? Which is like,
Yes.
yeah, but like, I think like obviously I am like a technological optimist, right? I think we will solve problems in human biology and medicine and many other things in biotechnology through the like learning about biology and then the application of technology towards them. And like you can take part in all sorts of ways towards those things, and they're really exciting. Whether that's like I barely do science now, if at all, right? Like I mostly just manage and and like manage people and like
Hmm.
kind of project manage stuff. And um so there's all sorts of ways to have contributions, and they're all kind of important. I I I think the second part is like I just really enjoy being a part of science. So like I think finding ways to do that over the course of your career, there's like all sorts of ways. I so I guess in the non-traditional advice stuff, I'd say like if you're not happy at some point, like go switch. Like I've switched many times in my life, and it's not about it's not about like the work. Like obviously sometimes you just have to slog through stuff, and I'm a big
Yeah.
believer that you do have to do that but finish things. Um but like ultimately I think that like you're going to get a sense of what's exciting. The way I do this with myself is when I stop being obsessed with what I'm working on, I tend to get obsessed with other things, and that's when I realize that like, oh, maybe I'm not like my head's not in this thing, and I
Yeah.
should go explore that for a little while, and then just staying open to opportunities around those. And that's kind of how I've definitely have played it over the course of my career. And I think just like I've switched labs in my undergrad lab, I've kind of left the startup to go back to academia. There's like a
Yeah.
bunch of different like places where I've done that. And every single place I've learned something new and exciting, and like those have been really useful things for my own career. Um, I think the third is like um, for young scientists, like your peer group is like basically in 20 years going to be all over the world doing all sorts of cool stuff. So like I think there's that that I didn't realize as much when I was younger, that like all the people that are the best people you work with are going to like be doing awesome stuff because they're probably really good, and like and so like finding ways to work with the best people you can, and work on the like the most exciting problems you can, especially early in your career, I think something you should definitely do.
Awesome.
Well, I think, and then I'd say the last thing is that in general, I generally think that like learning something, like when you're on a fast learning curve, it's generally really uncomfortable. Like meaning like it means you're probably bad at what you're doing, and you're learning really fast how to get competent because like, and so it's not always a great feeling. And so especially early in your career, you're like, why am I so bad at this thing? Maybe I'm just not good at this thing. But like it usually is just like you kind of just have to get good at it. And this is like you having to take the time to do that. And that like you don't want to get off of like I guess I'll put it a different way. Switching is not because you shouldn't do it because like, oh, I'm not good at this thing per se, or maybe this is like it's actually like I think people confuse the two, that like, yeah, you're going to suck at almost anything you do that you're starting out at, right? And so I think over the course of my career, I've gotten way more comfortable being dumb about lots of stuff.
And
And but
uncomfortable.
right, and really uncomfortable, but taking the commitment to be like, okay, I'm just going to get better at this thing. Like I'm going to learn the vocabulary. I'm going to like be able to like design good experiment or whatever it is, whatever part. Like for me, it's been like, how do I manage people? Like how do I manage an organization? Like, and so whatever it is you're learning in your career, it likely means that you're going to be bad at the thing when you start, and um, and that's okay. But like it, like I think a lot of people try to like quit at that time early, like rather than like saying like, oh yeah,
Persevere.
I'm learning a lot. That's why I'm really uncomfortable, right?
Yeah.
Um, yeah. So I'd say I don't know. Those are probably
I love it. I love it. And I think you're the first one who actually talks about discomfort and not and sucking at something that you just started doing, and being having the ability. I don't know if you can hear it, but my 1.5-year-old is screaming in the living room. I hope you can. About giving it a shot and living with that discomfort, because anything new that you're going to be trying out, you're there to learn. You have to learn quickly. It sucks having to do that. But once you become really comfortable in that situation, you get to a point where you look back and like, actually, that wasn't that bad. And look at all the skills that I learned, and you can take
yeah,
that to the next level.
yeah. I think it's super important, and also being around people that will tell you that you suck, in the
Yeah.
nicest possible way and the most
Yes.
supportive way, in a way that has built trust. But like you want to be in a place where people can give you critical feedback, right? Because that's the place you can learn the fastest.
I
And
think so too.
yeah.
You mentioned you were in July at the career panel of the ACR GPCR day meeting, and you had mentioned that your co-founder or someone you work with on the business side of the company had told you that after your first year that you sucked at something, and how
Oh,
much
my
you
like
learned.
yeah, like performance review, like
Yes.
yeah, no, it's horrifying to get real performance reviews.
It
But
is.
like I've learned to like really love that, right? And like I think I've been able to improve much faster than I would have been without somebody giving me like really critical advice, right?
Yeah, and I think you need to have those conversations as often as possible so that you don't let you can course correct. But it's also important to say, well, you're doing this really well, then this is the things that you need to improve on. It's kind of that proverbial shit sandwich. But I think it's also important to acknowledge what you're really good at in general. All right, Tree, we have 4 minutes left. Top three aha moments that defined your career as a scientist.
Top three aha moments. I don't know. I like I don't know if I've had like super big aha moments. Like there's definitely been ones where uh I think I was at my postdoc, um and I had boarded a flight, uh and I had like my cell phone ran out of batteries, my phone, my laptop ran out of batteries, and like, and I had downed this huge coffee, like a huge Starbucks coffee right before the flight, and it was a day flight, and it was wired in a middle seat, and I had nothing to do, and so like but like I think I came up with like literally 4 papers worth of work in that flight. Like basically, we had like 4 ideas that came about. I was like, why am I doing like? And like those 4 things ended up being like 4 papers out of the postdoc that like I think like, and some of the biggest work that led to from that one place. Like there's been a few things like that where it's just like I think mostly when you're away from distractions and like wired, and yeah, there's been a few like that. Like I think like during the pandemic of like kind of repurposing our platform, like some of the stuff around some of the targets and stuff we're going after at Octant. Um the and obviously some of the work with people in the lab and others like during my career. It's there are these kind of moments where everything starts coming together and it is really exciting, and
Yeah.
but it is like the 0.1% of the work. And many
It
of
is.
those like, and they're only exhilarating in retrospect because at the time they seem like crazy ideas, right? Like so
Yes.
like yeah, so I think there have been those things, but like if you look at all the things that didn't turn into cool papers, like,
But I think it's okay.
yeah,
I think that's true.
yeah,
I mean, writing a grant on factory GPCRs, getting the grant, but not getting the check, I think is a determining moment in your career. When you think about it, must
yeah,
have not been fun to learn that actually you were never going to get the funding from that grant, but you'll learn something.
yeah. Well, anyways, I've really been enjoying these podcasts. So
Thank
I'm
you.
excited that episode 94, you said.
I think so. I think 94. Yes, I think so.
Oh wow. Well,
All right.
that's super exciting, and thanks for having me on.
Of course. Thank you, Shri. Don't go anywhere. All right. I'll talk to you soon. Thank you for joining us and listening to this Dr. GPCR podcast episode. I'd like to thank our guest, our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and Montana Molecular. Thank you to our Dr. GPCR team as well, Attila Forrest, Ines Pinero, and our newest Dr. GPCR proteges, Montserrat Avila Zozoya and Nipuna Wirasinga. Welcome to the team, Monse and Nipuna. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any question and suggestions at hello@drgpcr.com. Until next time, stay safe, and we'll see you in the ecosystem.